So now what are we going to do with our messenger RNA transcript that's finished? Well, remember on the messenger RNA, as on any RNA or DNA, we have nitrogenous bases, those letters A, U, C, and G. Remember, the U takes the place of the T that's normally found in DNA. So on a messenger RNA transcript, the letters represent the nitrogenous bases of the RNA. And a ribosome is going to read them in sets of 3. And those sets of 3 are called codons. So let's just imagine I have a messenger RNA who has a codon AUG. What is that going to translate into? So we want to go from the RNA language to the protein language. Well, if we use our codon table, we can find what it's going to translate into. So the first base of our codon is an A, so I find the A area. The second base of my codon is a U, so I find the intersection between the A and the U, and I'm in this box. The third letter in my example was a G, so I find the third G. So where the A from the first letter, the U from the second letter, and the G from the third letter meet is right here. So AUG was my first codon on my messenger RNA. In green, I can see that it is coding for MET. So what does MET stand for? If we look at this chart, we can see MET stands for methionine, which is one of the 20 amino acids. It's also represented by the capital letter M. So now let's try another one. Let's say my codon is AAG. So my first base is A. My second base is A. So I'm going to go to where the A's of the first and the second base intersect. I'll be in this big box. And my third base, I think I said is C. Sorry if I didn't. AAC would stand for or code for ASN. And if we go to this table, ASN stands for asparagine, and that is represented by a capital letter N. So a codon table is used to decipher the code in the messenger RNA. The code in the messenger RNA is 3 letters at a time. Each 3 letters code for a specific amino acid, which is represented in this codon table by these green letters. So then what do these stop signs mean? If you get to a codon in your message that codes for a stop, so your codon would either be UAA, UAG, or UGA, that means you're at the end of your polypeptide. And your ribosome, your messenger RNA kind of disassemble or come apart, and you no longer produce a polypeptide. So again, here is an example of this was originally your DNA. Your DNA was transcribed into your messenger RNA. And what's missing from this slide is the messenger RNA was then modified, or basically the exons and introns separated from each other with the exons put back together. And now we have our mature messenger RNA. We can see we have AUG as a codon, UAC is a second codon, and UCA is a third codon. These are the amino acids for which those codons coded for. So the language from the DNA was transcribed into the RNA language, which was then translated into the amino acid sequence, which assemble in the correct order to make a polypeptide. The instrument of reading a message is a ribosome. And a ribosome is made up of 2 subunits or 2 parts, a large subunit and a small subunit. And Inside of this outline of the large subunit is what is believed to be the actual protein RNA folding of the large subunit of the ribosome. Same concept here, but we're looking at the small subunit. And then over here we have an artist's rendition of an intact ribosome, which will be found sandwiched around a messenger RNA molecule. Another part or another player in the translation process is our tRNA molecule. And we can see here, this is called a space filling model of the tRNA molecule. And this is a more simplified version. And in this more simplified version of the model of the tRNA molecule, we can see that on the top part, we have a sequence of 3 nucleotides. In this example, the nitrogenous base of one nucleotide is A, and then of the other 2 is C and C. This is going to be complementary to the codon found in the messenger RNA. And the messenger RNA has the code. The anticodon is found on the tRNA, which is carrying the correct amino acid that should be placed in our proper sequence on our formed polypeptide chain. So the codon has a complementary anticodon, which is found on a tRNA, which that anticodon is then carrying the correct amino acid. And that's where the codon table all comes in, with having specific codes on your mRNA match with specific amino acids.